Rahul, are you there?
Madam, starting recording starting.
YouTube live stream charges is okay? Okay, sir.
So, very good morning to all the bright-faced delegates. Over 1,000 plus. And very good morning to our plenary speaker, who is from International Institute of Biotechnology Division. And I think it's our great pleasure, sir, to have Dr. Pavan with us. And I think we are very fortunate through the connect of Tiwari, sir. We are able to have your message, and we are looking forward to know more about the cell technologies and the innovations happening. And I strongly believe it's an order of the day where either you innovate or there's no place for us to stand there. And I think the title of your team is also aptly used. And I think we look forward to hear many things happening. And for all this, I think I thank Tiwari sir and my team of people, Vanita, Santosha, and all the team who had the will to do it. And until will is there, I think automatically skills will come up. And we wish to learn, and we all are knowledge thirsty to learn from you, sir. Thank you, and over to you, sir.
Over to you, Santosha.
A very good morning to all the dignitaries present over here. Dr. A. Ramesh, who is the chairman of the event. Dr. O.N. Tiwari, sir, who is the co-chairman of the event. Dr. K. Vanita, who is the convener of the event. And Dr. Pavan Juturi, who is the resource person for today's event. I welcome you all for the second day of 5-day FDP program on advances in science and technology innovations. I'm Dr. Santosha, and I'm greatly pleased to introduce today's resource person, eloquent speaker to you all, who is none other than Dr. Pannaga Power Jutu sir. He has completed his MSc from Kuvempu University, Karnataka, MPhil and PhD from Pondicherry University, Pondicherry. And Dr. Juturi is currently group leader, Omics of Algae group at Icgb, New Delhi. His primary focus of research is understanding the dynamics of microalgal system through integrative multi-omics approach in coherence with reconstructed genome scale metabolic models with well-defined functional pathways that will elucidate an effective strategy for converting light or carbon source to biomass, biofuels, and biorenewable for sustainable futuristic solutions. These findings will provide important breakthrough on central metabolism in these microalgae, which are required for biotechnological improvement of next generation biofuel production. His today's topic for discussion is self-factories, new frontiers and innovation in the era of industrial biotechnology. So this is in brief about introduction to Dr. Pavan sir. Sir, I request you to take over the session. Over to you, sir.
Thank you very much. Can you make me the host or co-host, please?
Sure.
Yeah. Already you are the host, sir.
Okay, now it is. Yeah. Okay. Okay, very good morning to all of you. And first of all, it's a great privilege and honor to share our research today. And I would like to especially thank VIPER organization, especially Principal Dr. Ramesh, Dr. Vanita, and co-chairman of this symposium, Dr. Tiwari, and all the people who are involved in this conference to give this opportunity to share what we are doing at Icgb. So basically, I think you are all able to see the slides starting from here. So I'm trying to show you in the background a very popular protein called as Rubisco. Without that, we will not be able to survive on this planet. We know that it is fixed, carbon dioxide is fixed by Rubisco, and it is oxygen is given out, and that's why the highly evolved human beings or other species on this earth. So this is just a pictorial. I'm not talking anything on Rubisco today, but I will be highlighting the basic niche of our group, what we are working at Icgb. And mostly they are, see, I believe in, if you know the basic biology of any cell, then you can transformate that cell into genetically modified to produce any kind of, that is the requirement of future. And being a budding scientist or budding students who are going to take up in this area of industrial biotechnology, I would like to emphasize more upon the relevance of how techniques can help in understanding. Basically, what this thing gives you, that an understanding what is happening within the cell. With the naked eye or with our eyes, we cannot see what is happening within the cell. And a lot of metabolic processes, or what we call it as the metabolic regulation, happens within the cell so that it can control what it has to produce and what it has not to be produced. So in order to understand, we understand the basic biology or fundamental science behind that is through omics as a tool. And for this, we are today, as I try to crunch all the data into 1 hour lecture so that you can always have the glance of what we are doing at Icgb. As I told that I welcome all, and this is my email address, and I'm happy to interact with the students and collaborate with the faculty if they are interested in near future. So with that, I once again thank all the committee members of this wonderful FDP program, and I would take you to the next few slides telling about what is Icgb and where we stand. So Icgb, many of the faculty know where Icgb is located, but to know for the students, especially Icgb is an international intergovernmental organization, especially involved with research, training, and technology transfer in the field of life sciences and biotechnology. And we are located in 3 places. Our headquarters is in Italy, Cape Town, and I am presently located here in India. And on my picture background, you can see the campus. And one more campus is in South Africa, where we do activities in different pertaining to life sciences and biotechnology. So we provide PhD program and postdoctoral programs. We don't have master's program at Icgb. So we have a purely research organization where we work on different thematics. So we work on cutting science research, where we train different short-term PhD students and postdocs. And we also conduct different number of meetings and workshops, just what you're doing it internationally for developing and underdeveloped countries. So also we have many access to the state-of-art facilities, and we are well equipped with best of the equipment in the world. And also we are instrumental in achieving sustainable development goals of 2030. One of the primary relevance of our Icgb is to also. So on to my right, if you can see, these are the core areas of research, what we do at Icgb, like infectious disease, non-communicable diseases, medical biotechnology, industrial biotechnology, plant biology, and biotechnology. So where I belong to industrial biotechnology area, where we are one of the basic group which try to understand the fundamental science of algal omics. What is that? You will understand when I go through my presentation in due course of time. Basically, I work with industrial biotechnology, biofuels area. So with this short introduction of Icgb, I would further take up what are the things I'm going to cover today in a nutshell when we're talking about biofuel generations, why we need the biofuels and classification of algae. As our eminent speaker yesterday, Dr. Tiwari, was talking about different types of algae. So I'll just put on, and I'll be concentrating on area number 5, which is called omics of algae group. It is a very unique field in the world, and we try to work on different layers of algal or research, employing omics as a tool. So what is that? I will be focusing on that. And basically, finally I'll end up with whether what we are talking about biodiesel or the future substitute for fossil fuels is possible. Yes. So that is what an example. The road to $7.5 per gallon is the one which I'll be summarizing, and also as I'll be highlighting many things which is related to pharmacopoeia and nutraceuticals also. So first, to understand, especially this is for the students, mainly to understand the biofuel generations. They are majorly 4 generations of biofuel. This is presently are there, but here you can do first generation, second generation, and third generation biofuel, and fourth generation is the CO2 or this thing. So I majorly work on third generation biofuel, which involves algae as a model system. So the tools what we use is universal. You can use in mammalian system, you can use in plant system, you can use in pharmacopoeia, you can use in anything, because the tool, analytical tool we employ in the lab is omics tool, which is universal, and the future of anything, like you take medicine, you take plant biology, agriculture, you take algal research, or you take biofuel research, everything is involved with the omics research. So without understanding the fundamental science, we cannot manipulate to produce the byproducts, so or the metabolites of interest. So to brief this, how these 4 generations are, the first generation is with sugarcane, rice, wheat, where first generation biofuels are edible crops. And there is no doubt we are not going to use any of this first generation as a substitute for biofuel, because there is a huge debate. This is food versus fuel, and food is equally important as a fuel. So we are not using to have edible crops as a raw material to generate biofuel. Well, the second generation is lignocellulosic biomass. You know that sugarcane bagasse, forest residues, cell biomass from fermentation. There are a lot of byproducts. So for example, you have a sugarcane bagasse. So you have a sugarcane, you remove the sugar component, and you have the bagasse, which is of no use. So we can use that raw material to generate bioethanol. And the mandate of government, even government of India, is to blend 20% ethanol by 2030, which is a mandatory for most of the biofuel where they're working on bioethanol. And many of the biofuel refineries are establishing these bioethanol plants in India, like HPL, HPCL, IOC are all involved in establishing these biorefineries. The third very important where we are looking upon is third generation biofuels, especially algae. You can name any kind of algae. Each algae has a specific importance. Either it produces biodiesel or other high value products. What are the things it can be done, and what is the thing? The beauty of algae, as yesterday Dr. Tiwari was discussing, that it can produce multiple products, and how you can utilize that product to produce very high economical value of that. So in a nutshell, third generation is to algae, and the fourth generation is non-edible or genetically modified, sequestering CO2, Jatropha, and all those things. So basically, you can see the 3 layers where you need raw material technology to transfer the selected raw material into particular compound, and finally the products like bioenergy, biofuels, or other things. So basically, first generation, second, third, and fourth are the raw material selection, where you select the particular raw material. If you are selecting the second generation, you get ethanol. If you are selecting third generation, you get the biodiesel. So you require diesel as a biodiesel as an alternative for the fossil fuels. By 2050, most of the fossil fuels get decreased. So we know that we require an alternative source. So the problem what we are working is the tomorrow generation problem. It's not for the present this thing. So it is very important to solve and have in our pocket the solution for this, where we can have an alternative source for fossil fuels. So we are working next 50-year project where we can look upon what will be the next generation for looking for. So I'll not be discussing much on that. This is like when you have a raw material, how do we extract those using physical means, thermodynamical or chemical or biochemical, that can be converted. And this is where the engineering groups come into role, where they can design different bioreactors, and the products can be formed like biofuel, jet fuel, and all different things, along with bioplastics, food products, everything. So This is how, in a nutshell, if I want to explain the biofuel research or biofuel generation, this is how it looks like. Okay, so as I said, like fossil fuels, why do we need biofuels? Fossil fuels are limited. We need an alternative source. Global energy demands keep on increasing every day to day in the world, and CO2 release from the fossil fuel causes climate change. And the positive thing about biofuels is the CO2 can be sequestered, and that it becomes a carbon neutral. So we are decreasing the climate change, or what we call environmental this thing. And liquid petroleum transport fuels are hard to replace by any other renewable sources like wind, solar, and hydro. And this is the need for the hour. So we cannot start when the problem arises. This is required a solution very ahead, so that when the problem arises, we are ready to take it up further. So just to brief, yesterday we were talking about cyanobacteria, algae, and microalgae. These are different groups of algae, and each algae has its own importance. So this is what we call as a green gold, because this is what we have not till now explored in certain ways. And algae is present everywhere. So you see the ponds, it is full of algal biomass, where you can see the green matrix on that. And you usually don't go into those waters, but actually that is a treasure what we have in our, and India is a huge coastal belt, and we have, as Dr. Tiwari was saying, we have a rich culture collection in and around our natural habitats where we can look upon. So these are some of the few examples of cyanobacteria. It may be filamentous, single-celled, prokaryotic. Cyanobacteria are mostly single-celled organisms, followed by microalgae, which is the one where we work majority. It may be a Dunaliella, it may be a Spirulina, it may be a Chlorella, it may be Chlamydomonas. Different types of microalgae is available where you can produce different products. The red one you can see is Hematococcus or Petrococcus, where you can produce astaxanthin, a very high relevant pharmaceutical compound in the having a global market. So we have diatoms which produce glucoxanthin, carotenoids, antioxidants, nutraceuticals, whatever you name, even the omega-3 fatty acids. We are in a perception that what the fish we eat, the omega-3 fatty acids from the fish is what we obtain is from the fish. No, because fish feed on algae, and indirectly that omega-3 fatty acids get accumulated within the fish. So the basic source of production of omega-3 fatty acids, which I'll be covering in my last part of the lecture, is algae, microalgae. Final classification is macroalgae. It's very popularly known as seaweeds, where if you are if you are fond of Japanese cuisine, you can find the seaweeds as a part of their cuisine, and it's very popular. So you can use as edibles. The primarily these are carbohydrate producing algae, so it is rich with carbohydrates, and this can be a source of second generation biodiesel as an alternative other than lignocellulosic, because they can be produced in huge biomass. And what we see in the oceans and seas as a big islands is of corals. Along with the corals is seaweeds or macroalgae. So that's this is the relevance, and each one has its own importance. And the color represents what kind of pigment they contain. Rhodophyta has red pigment. Podophyta has they have a brown pigment in that. So that's how the depends on the carotenoids and the pigments which are present in a particular algae. These are classified. So I'll not go into anything into the deep into the physiology, as yesterday Dr. Tiwari was covering the physiology, the general aspects of how you can identify the algae and all. So as I told, why algae? Algae can produce huge biomass than any agriculture crop or oil crop per productivity per acre. It can be used in non-arable lands and wide variety of water sources. You can use in wastewater, you can use in marine water. No need of fresh water to grow the algae. So it can be used in non-arable lands. And also, as I said, it can be used in mitigating greenhouse emissions, which is a major concern in highly populated cities like Delhi, Hyderabad. Pollution problem is the highest, and the sequestering of CO2 and greenhouse gas emissions by algae is very important, and it can go. So the basic beauty of microalgae or algae is it has not only produced biofuels, but also other high value co-products, which can be simultaneously extracted along with the production of biofuels, which adds to the cost. That's how we can make future biofuels sustainable. So what are the challenges we usually face in large scale cultivation and harvesting? These are the two major issues in algal biofuels. And that is why we are not into that level of industrial. And these are the two major costs, which is producing algae at a very high cost. So we need to produce a barrel at around $100 and $150 so that we can compete with the fossil fuels. So these are the different areas where you can improve upon, and people are working on. And these are the challenges which has to be addressed to bring down the cost to the comparative fossil fuels or less than the fossil fuels. To start with a lecture, I would like to show a small video here where this video shows, it's from ExxonMobil, the beauty of microalgae, why we should consider algae as a source for next generation. So this actually corresponds to what exactly we want to say with terms of algal research, especially. And that's a beautiful, so it's available in YouTube also. It's a beautiful representation of, I am very small, but I can do many things. So that's the algae wants to convey to us. Even I am very tiny. It's not even small. It says that I'm tiny, but I have a lot of things to do. I can do very this thing. So it brings the hypothesis of why the algae is so unexplored till date and what is the relevance now. And now it is the right time to understand fundamentally and translationally. That means both it has to be worked on multi-dimensional approach, or called what we call it as multifaceted applications of microalgae. So we call our group as B3 algae or omics of algae. B3 represents, you can see, biomass, biofuels, and bioproducts. So B3 represents the three Bs, and it's very important because it utilizes three. It's like a computer programming. So algae is the CPU, and there is an input and there is an output. So the three major inputs for an algae is sunlight, which is very much essential. Carbon dioxide, it may be industrial wastewater, waste gas, or atmosphere, and it can grow in any kinds of water, that is wastewater, freshwater, seawater, and water is required. These three components are required with the minor nutrients addition, and it can produce biomass, biofuels, and high value products, along with other important relevant molecules like hydrogen gas, which is very much now biohydrogen is gaining a lot of importance from government of India to have fuel cells using biohydrogen. And electricity also can be generated. We had recently a paper with Amity University in collaboration where we have showed how it can produce bioelectricity using microalgae, not only with fuels and hydrocarbons. This is a very interesting area where we can do bioplastics as a major product output. So these are the products which are coming out of algae. You can, from the biomass, you can use an animal feed, fish feed, biofuel. You can use a generation of electricity, and biodiesel and jet fuel can also be obtained from that, and high value products. This is where the pharmacopoeia and nutraceutical products come into major role, like antioxidants, vitamins, omega-3 fatty acids. So this is a time to understand the natural products, how you can compose your pharmaceutical products or drugs with the natural products so that it can be directly consumed. And also, as I said, like it can be used in fuel cells, photovoltaics, and also bioplastics. So that's why we call it as B3 algae, and we work on all the three major components of that. And how do we work on that? That is what I'm going to discuss in three different components: how we can increase the biomass, how we can increase the biofuel and byproducts. So that is what our major goals we are trying to look upon through fundamental science. So fundamental science is equally important to understand how the organism is behaving with a particular condition. So future perspectives, this is recently a review article which came in Frontiers in Bioengineering and Biotechnology. This is not from our group, but I've picked up a summary of the work which has been very interestingly demonstrated. So you can see onto my graph, this when you have a strain, promising strain, you do multi-omics and gene mining to understand how it is. You establish a genome scale model of that, and using the mathematical fill in the gaps and construct a high metabolic model. So you don't require to do experiments. You can do in silico and understand the cell within the cell. And then you can do the pathway targeting, or what is called metabolite targeting, through regulating different gene transcription disintegration through CRISPR-Cas9. And that when produce the condition specific omic data sets, which you can compare with that, and how the flux is being diverted. That means if you're giving a nutrition, how it is going towards that, and you can make an engineered strain that you can control by itself, and that's how it can produce a what is called an industrial cell factory. We call it as photosynthetic cell factories or microalgal cell factories. So that's how you can have a product predicted and target specific cell factory. To order to understand, we do most of the work here at Icgb in different layers, and the major tool what we use is omics to understand the fundamental science. So this is our thematic, what we call is new frontiers in algal biotechnology, where I told but the major concern is biomass, biofuels, and byproducts, which you can see in the center. And see, for any, this is especially for students, if you want to understand any system at omics level, your experiments should be very accurate to understand those things. So you cannot just put in the media and take it out from that, and you don't have a time course experiment. You cannot reveal what is happening with the due time course. For example, if a cell can take within 24 hours the fixed carbon dioxide, that carbon dioxide goes within the cell to different pathways to make biofuel. So how much duration, that's called a time course, to fix the CO2 or any kind of compound into a valuable product, that's the time course. So it is very important to design your omics experiment through time course, either starting from 0 time point, and it may be 10 days, 9 days, or it may be 20 days, depending on the product, what you're looking for. So the two scenarios, what we look at is two sides of a coin, just like either you can do nutrition deprivation and increase the biofuel, and genetic manipulation, you can increase the biofuel, but unfortunately, the biomass gets reduced. So we have developed a new concept of media engineering, where you can just, so naturally these strains are producing the biofuels and byproducts in nature. And the fossil fuels which were accumulated from the centuries are nothing but the debris of the sea belt of algae, which is being settled down in the oceans, deep oceans. So naturally, these are able to produce these products. How do you do that to enhance that? That's what we are trying to look. That is like media engineering and chemical modulators. One interesting concept where we try to develop through media engineering and chemical modulators is we don't compromise on the biomass. As I said in the first phenomena, we compromise highly on the biomass, but we don't compromise on the second part, the biomass, but able to produce the byproducts and the biofuels in a substantially higher amount. So in order to understand that, what are the tools you require? You need molecular tools to understand the fundamental biology. So you cannot just do what is happening within the cell, and to analyze those cells, you need different high-end techniques within different strains. So to understand that, the one major tool what we use is omics research. Omics is nothing but genomics, transcriptomics, proteomics, metabolomics, fluxomics, all together comes as integrated omics. And that is what the powerful tool we use to understand the fundamental science, what is happening within the cell, which cannot be visualized by any instrument. So we are trying to establish and to build a cell within the cell. So technically, we are trying to understand what is happening in the cell, rebuild what is called reconstruction of the whole pathways, what is happening within the cell, and to understand and then manipulate. That is why you can manipulate with genome editing or CRISPR, where I'm not covering today, and synthetic biology. So you want to produce something through synthetic and system biology, no need of doing all the experiments, but you can do in silico, that is computational tools you can use and generate a model which is better, then try to manipulate a particular pathway or a gene of interest. And pathway engineering, it may be a transcriptional engineering, it may be a gene engineering. So we came up a new, so previously all like this before 5 centuries or 6 centuries, people used to target a particular gene of interest and overexpress that and produce the product of interest. So nowadays, we can control the transcriptional factors or regulators so that you can overexpress those transcription factors instead of nothing to disturb with the pathway engineering. So we can just try to improve the transcriptional regulation of the particular transcriptional factor so that we can enhance the byproduct. So that is called pathway engineering, which is a very new, or this is also known as transcriptional engineering, other than gene and metabolic engineering. So these are the various tools what we understand, we take up in the lab to understand what is happening within the cell. And that is how fascinating and a novel research approach what we take upon to understand the fundamental science of the algae. So as I said, I will be covering 3 different topics like biomass, biofuel, and byproducts as a separate section. So major concern, as I told, that we can improve the biofuel, you can improve the byproducts by doing different conditions, genetic manipulation, nutrition deprivation, but you cannot improve the biomass. So we cannot compromise on the past. So we came up with a strategy, and these are the few publications last year and couple of years. Along with some chapters, we try to publish our work, and these are highly reputed peer-reviewed journals. By name, everybody knows these are very highly rated and cited impact factor journals. So, we, in order to understand what we have subjected the CO2 as a means of carbon partition, photosynthetic carbon partition to understand. So we employed as I said, we should be technically strong in designing our experiment. Otherwise, after 3 years, you do all your omics experiment, it goes wrong, everything goes wrong. So it is very fundamental that you design your basic experiment very strongly so that what is your focus of your research, what you are going to get it is very much need to be understand before you start an experiment, because these are very expensive experiments and time consuming experiments, and a lot of labor, hard work will be involved with even one single experiment. So basically our rationale here is to understand the cell dynamics of any cell factory subjected to carbon supplementation using multi-omics approach to identify the enzymes and also carbon concentrating mechanisms that will contribute towards engineering of regulatory networks for increased cell biomass. So this is what I have already explained, and this is what you are looking for, how to improve those yields, cell biomass, or not compromising the cell biomass. So to understand that, this is recently published paper and it's available online. And we, as I said, we subject to very low concentration CO2, high concentration CO2. What is very low concentration? It is air. So you in air, it is present at 0.02 to 0.03%. Volume by volume of CO2 is present. At high, it may be 3, it may be go to that. And some species are able to even take 20% CO2. But our strain, this is Microchloropsis, it's a marine strain, and it is very good at 3%. So we are fixed at a 3% as a high carbon concentration where it can. So to understand, and it's a non-pyrenoid based. See, it is very important to know that all the chloroplast or rubisco are located in the chloroplastic, so pyrenoid-based system. And this is a very unique and it's a very primitive strain. That's why they are not non-pyrenoid, and how they have developed these rubisco or CCM mechanisms in order to fix the CO2 even at very low carbon in the deep oceans is what we are trying to understand. To design, we can see that at very high concentration, our growth profile increases, our chlorophyll, carotenoid profile increases, the biomass, everything goes on well because we are supplementing the better the source. So that is nowadays it's very unique that that's what is called as media engineering. The amount of carbon and nitrogen you supply, the better it grows. So that is what the principle we are looking and what happens to the metabolic pathways. So in order to understand that, we apply an approach called Metabolomics Approach. There are two approaches in omics, which is called just like forward genetics and reverse genetics. I don't know whether you have covered in your biotechnology course, but reverse omics is the one where you don't have anything on that data, genomics or any transcriptome. So you understand from the metabolite level and go towards proteomics, transcriptomics, and genomics and try to integrate and know the knowledge. So in order to understand that, we do a reverse approach where you can use liquid chromatography or gas chromatography along with mass spectrometry. And this is the whole setup. You have the samples, you remove the protein, you take the metabolites and do derivatization and run through LC and MS or GCMS and analyze through that. This is a simple cartoon how you can depict the metabolome. And from that metabolome, what you can see is onto your left side, the number of metabolites. And this is especially employing GCMS. And you can see their number of fatty acids, sugars, amino acids, organic acids, which has been substantially increased. And this is called time course. So we are trying to understand from 0 day, 3rd day, 6th day, and 9th day, what is happening within the cell. You can see from this, it's very important to see what is happening is alpha linolic acid, which is a precursor of omega-3 fatty acids, is increasing in the 9th day. That means that is how you have to understand each metabolite. So you have hundreds of metabolites, and it is usually metabolites will be in hundreds or thousands, whereas transcripts or proteins will be in lakhs. So it is very what we call it as big data or huge data, which has to be in silically analyzed. So this we are showing how many number of metabolites which are upregulated and downregulated, and this is how we can. So based on this, we come up with a hypothesis where what happens with the VLC and HC condition. So onto your left, you can see at very low carbon concentration, mostly the carbon is fixed as a carbohydrate molecule rather than a lipid molecule. So onto your right, if you can see, then there is a high carbon present. So that carbon is diverted towards multiple products like lipids and alpha tocopherols and all these things. So that's why when there is a maximum amount of carbon and nitrogen ratio, the products formed will be maximum. So then in that way, your biomass will not be compromised, and the products you can, and also simultaneously, you can produce multiple products at a time. So we have further to understand, because CCM takes place within 24 hours. We want to know what is happening at the proteome level, and this is just typical to same thing metabolome level. So we try to understand through the bottom-up proteomics approach, where we have isolated the proteins, trypsin digestion of that proteins, and analyze those proteins. So you can see number of proteins which are upregulated and downregulated within HC and VLC condition. Again, I'm showing you between 0 hour, 6 hour, 12 and 24 hours, where the number of proteins which has been upregulated and downregulated. So this has to be, I'm just rushing through this because it's just a very sensitive data. And in order to understand, you need a more knowledge on mass spectrometry and this thing. So basically what we want to say is, through our metabolic profile, we can underlay what is happening. And these are the metabolic pathways, what we try to establish. So this represents a schematic representation of carbon partitioning, which is happening between the very low carbon and high carbon. So the dark, the white represents 12 hours, 16 hours of photoperiod with 8 hours of dark, light versus dark. So what is happening within that, and what are the mainly to understand. The green is the metabolic pathways which are upregulating during very low carbon concentration, and the blue ones are the one which are highly upregulated during high carbon concentration. As I said, the TCA cycle will get enhanced, and there are a number of products which are formed. So through what we've spoken on metabolomics is being actually reflected in proteomics. That's how we integrate those to understand. So basically, this is a schematic representation what happens, and now we can understand within the cell. So you cannot see what is happening within the cell like this. So we make it to understand, in order to understand, okay, this pathway is upregulating, this pathway is upregulating, and now you can control the cells in the presence of the particular concentration of CO2 and particular this phenomena happens. That is how you are trying to understand the fundamental mechanism of these cells. So the second part is biofuels, where we are trying to look upon the biofuel precursors, that is biodiesel. And these are the major research papers in these couple of years from our group, along with few chapters. I'm just highlighting very important papers, not all the ones, but I think these are available online, and you're free to go through that. And if you have any queries, come back to us. So, and also I'm happy to share that we have done a hybrid genome assembly, the first of its kind for any microalgae, and it's completely done in India and completely done at Icgb with all our in silico in-house tools. So only thing is we have sent the sequences for Illumina and Nanopore to get the sequences done, but all the assembly part and annotation part has been done at Icgb. So I'm happy that we have been successful in laying a pipeline how to do different analysis if you have the omics data set. So, so as I said, a good experiment will give you best results, and how do you design your experiments is very important. So understanding your cellular dynamics. So you want to understand the biofuel improvement. So you have to give a nutrition stress. It is a well-known phenomena, and many people have worked on that. So we give a nutrition stress and see how this done. And this is one of the projects where we worked on end-to-end. That means we have done on genomics, transcriptomics, proteomics, and metabolomics, and have an integrated omics platform. So many papers say that they have done metabolomic, proteomic, or metabolomic genomic. They call it as multi-omics or integrated omics. Anything more than 2 omics is represented as multi-omics or integrated omics. So this is the rationale where we want to enhance the biofuel. What we have done is, so this represents, so you have nitrogen deplete conditions. Within the center, you can see that is orange fluorescence. What we are seeing, that is nothing but lipid. The cells get bulged with a lot of lipid, and the tag bodies get accumulated. So this is a typical example where lipid biosynthesis goes up and photosynthesis and beta oxidation comes up down. When it is in normal nutrition, lipid biosynthesis is lower, photosynthesis and beta oxidation is higher. So this is a phenomena what we call it as stress biology in microalgae. So to understand this stress biology, we are subjected to different conditions, and to nutshell, we have seen that in nitrogen, it accumulates a higher amount of saturated and unsaturated fatty acids, which is important. And being the usually previously what the phenomena was like, if you have more saturated and more unsaturated, that is preferred for biodiesel, because the more PUFA you have, that will be leading to what we call as coagulation or solidification of the algae. So that is why we need less amount of PUFA and more amount of saturated and monounsaturated fatty acids. So keeping in this mind, once the hydrothermal liquefaction came up, you don't require. So you can produce any kind of lipids, irrespective of what you're looking for. And that can be broken down into simple precursor molecules, which can be used as a biodiesel. So again, here we have done a reverse omics approach to understand, and this is called targeted and untargeted metabolomics, where you don't know anything about, you do an untargeted whole cell metabolomics to understand, you break them into metabolites, you understand, validate through MS standards, and global metabolic profile of the pathways are established. So this is the typical example what we see previously, but this is for biofuel. You have number of metabolites which are upregulated and downregulated. And from that, we have hypothesized that The red represents the metabolites which were been upregulated due to nitrogen deprivation, and P represents phosphorus and S. So the red lines represent these pathways are highly upregulated, and the blue represents these are highly downregulated during the nitrogen deprivation conditions. So now we know that what kind of pathways are downregulated, what kind of pathways are upregulated. So we will be careful when we are doing genetic manipulation of a particular gene or a pathway in order to produce or bypass the carbon flux towards that. So further to understand, we done a proteomics, like previously I was explaining. We done a transcription digestion, and the peptides are identified, and the number of peptides were identified were nearly 2000. Proteins were identified, and the common between different conditions. Again, I told we do a time course experiment where we capture N+ and N-, which is nitrogen replete and nitrogen deplete condition. Significantly, there are 166 proteins which have been upregulated and downregulated. So significance is highly important. And I say that if it is 100 proteins, it's a very huge number to manipulate those proteins. So it is very important to understand what is happening. So next step, we further went into transcriptomics. I'm not dealing, so different kind of platforms are available in transcriptomics, Illumina and Torrent Solid to understand. So basically, we pull out the transcripts, and so you know that from the gene, the transcripts get upregulated, the transcripts are formed, the transcripts are translated to proteins. The proteins are give the instructions to form the metabolites of interest. So we are trying to map it from the bottom. This is the indigenous strain, and nothing information was available. So we have done from end-to-end technology, and we have sequenced everything. So this is overall, the red represents what are the metabolites which are upregulated and downregulated. And we have integrated the genomics and transcriptomics to understand what are the different pathways which are involved within that. And this is the in the insert, you can see red and blue lines, where these are different transcripts which are involved with upregulation and downregulation. So you can knock out this so that it will not affect any of the cell listing. So if you want to overexpress these proteins or genes of interest or transcripts, then your biofuel can increase. So this is how we can depict what is happening within the cell. So, and in a nutshell, this is how the hypothesis or schematic representation, what is happening. You can see the red pathway when it is subject to nitrogen deprivation. So the red pathway is highlighted, and that is one which is upregulated. And the classical blue pathway like TCA pathway and other pathways get downregulated, and the black represents no change. So this is how the pathways are regulating, and this is within the hell and nobody can see. So this is how through analysis of fundamental science through different mass spectrometry and transcriptomics and sequences, we can unveil the metabolic pathways, what is happening within the cell. So this is the first indigenous marine microalgae, Parachlorella castellari, which was completely sequenced. Around 300 scaffolds are present in that. Around 12,000 functional genes are present, and this we have already published in Algal research. And the pink here represents the fatty acid pathway, which is highly conserved in this pathway, and all the other pathways are missing. So this is how you can, these are the pathways which are present in the microalgae, Parachlorella castellari, and now you can manipulate according to the interest what you are taking upon. So we integrated all the data through integrative approach, and we can see this is how the cell looks inside. It's a very messy figure where you can understand, you cannot understand as a layperson, but they are integrated where the proteomics and metabolomics, transcriptomics, the yellow dots or bigger dots show the what kind of metabolites are upregulated or formed, and blue and red and other lines represent the genomics, transcriptomic and proteomic. And we have integrated the transcript and protein data so that you can know what pathways are upregulating and downregulating when it is subject to particular condition. This is how you answer the fundamental question, and this is yet to be get published. And this is the first of unique multi-omic data set what we have generated. So this is the thematic. What we are trying to understand is you try to understand through multi-omics approach. You put that multi-omics into, you reconstruct the pathways to understand what are the genes or metabolites which are upregulating and downregulating. And finally, you can put in the systems biology model, refine those models, and you can generate the microalgal cell factories, which you can control by yourself. So understanding biological systems to integrate it to engineer cell factories is the need of the hour for next generations. And so it may be anything. It may be pharmaceutical, it may be medical, it may be, see, the COVID. So what is the solution? Genomics is the solution. So without omics, nothing can be. So in future, personalized medicine also is to be do. So they look up your gene, they prescribe you what kind of medicines they have to give, they have to inhibit. So pharmacopoeia also will change according to that in future generation, to by knowing the genomics, by knowing the transcriptomics, what kind of drug can inhibit this particular pathway so that the cancer cannot cause. All these things, personalized medicine will be through this approach of, and even there is a beautiful study through omics where when astronauts go, NASA is conducting this study where they are trying to supplement with different feed what they usually eat and how it is metabolized within the body to understand what happens when they go in the space. So because they go in the space for 1 year, 2 years, so they eat on different stored fed, and one of the primary food for protein there is single cell protein, spirulina or chlorella, where they can use as a protein supplement. So it's a beauty where you have in every field, there are toxic algae, there are non-toxic algae. Toxic algae is the blooms, what we see in the oceans, and a very major problem that also be whenever the algae is produced high and it has to be eradicated, then that is called toxic algae. So you have different problems. You have beneficial and harmful, both has to be dealt with that. So when you have positive, we have negative. So in a nutshell, this is what the thematic, what we are trying to understand through integrated omics, and which gives a very clear picture of what we are trying to understand to engineer the cell factory. So this is the third and final part of byproducts, what I am talking about, biorenewables. So very important biorenewable, it may be omega-3 fatty acids, astaxanthin, fucoxanthin, it may be any kind of carotenoid, what we are looking for. These are very important and have a lot of pharmaceutical and nutraceutical approach. And these are our latest publications, which we try to, and this is the first of unique of its paper where we try to report tocopherols in an indigenous species where it is able to produce better tocopherol content, and till date, industrially relevant tocopherols are being generated through plant-based systems, but algae can produce more than that. So this is a proof of concept where the tocopherols can be. And this is one strain where Bifidobacter you can produce other high-value hydrocarbons, and also orange chocolaterum, which can produce DHA, that is omega-3 fatty acids. So these are the few publications which have been recently from our group. And again, the rationale here is to vary different carbon concentrations and understand through multi-omics approach how these omega-3 fatty acids are formed to produce different biorenewables. So we know that global market, expected market is around $3,400 million by 2025, which is growing at 4.2%. And these are different companies already present, and we are fortunate that a couple of companies are from India also. A major is Paris India Limited, which is producing algal byproducts. And these are very popular companies which are producing byproducts from algae. And these are the key players of global market, and more are yet to come up. But they are already doing it, and they are producing the high value products like omega-3 fatty acids, astaxanthin, and all other things. So these are different products of astaxanthin, which are beta carotene, omega-3 fatty acids. You can see the whole marine algae containing EPA and DHA. And if you see a little champs, Bonvita, nothing but it consists of DHA-rich oil from algae. The children usually consume the DHA-rich enriched Bonvita, which is present of nothing but DHA from microalgae. So there are many products, and many huge market is there. So if you can substitute this market with biofuel, that can bring the cost down of that. So to understand that, there is a unique heterotrophic microalgae which we try to supplement with carbon sugars. And our presence is because glucose will not be available for fermentation, it goes for fermentation. We want to understand what are the other simple sugars we can use. And in that context, we tried different sugars, and glycerol was the one which is able to produce equivalently or even better than glucose. So this is picture represents, so in glucose and glycerol, we have high amounts of omega-3 fatty acids, which are present. In others, we have very low, but there is a huge compromise in the cell growth. So again, we went through reverse omics approach to understand what is happening in this phenomena, and we found out there are different metabolic pathways involved in that. Mainly, there are two metabolic pathways which are involved in DHA production, which is a classical fast pathway, and the other one is the alternative pathway where you can produce DHA through that means. And you can see the arrows represent these are the different metabolites which has been enhanced in the presence of glycerol. And this is a very unique strain in the sense, like usually whatever strain it is, when you put in glucose and glycerol, they first consume glucose. But this strain consumes glycerol faster than glucose in the presence of glucose. We are trying to understand why that phenomenon is happening, but this is why our colleagues at ICT Mumbai have been working on this, and they were successful in combining two like omega-3 fatty acids from this thing, and also other strain to produce more, and they have used other different substrates to grow these things. So this paper is also available online, and that is how we try to understand. We have done multi-omics of this. Now I'm not presenting this approach, but this is how we try to assume different products. And the tocopherol story also we are not showing, but we have enhanced the tocopherol content or carotenoid content using media engineering approach in the presence of carbon supplementation, where we don't have any compromise in the biomass. So this is the paper what we published in Chemical Engineering Journal, which is the impact factor around 13. So this is the concept where we try to show that we can produce biofuels, biomass, and biorenewables. And this is a very pharmaceutical. So there we used a small molecules called chemical modulators. Basically, these are the inhibitors of cancer. So they act as a promoters in terms of microalgae. So basically the concept came like that. These are small molecules which can enhance the biomass, which cannot compromise but can produce. So we are trying to understand. It's a unique phenomena where the physiology does not change, but the metabolic regulation changes, so that within the cell, which triggers the cost-effective and sustainable algal biorefinery. So this is the concept of B3 algae on a whole, where you can produce byproducts along with the biofuels. So this paper is available online, and this is published in very prestigious Chemical Engineering Journal recently last year. So with that, I'll be ending my last couple of slides where I will be showing the road to. So I've told that many people have in their mind, is it biofuel really a myth, or we are just hyping those things? But if you see this roadmap pathway, it is very important that it is possible. So you, as I said, like you need to develop these things, and you have to have a co-product as a thing that can make it sustainable. So this is a very relevant that you can produce a gallon with at around 7.5 per gallon. And this is a roadmap, and this report is available, which is published by a huge group at USA NREL. It's called NABB report. And if anybody is interested, you can go to it. So this shows that the road to produce biodiesel is feasible, and we have to achieve by achieving all these problems here stated. And that's how we can make the biofuel sustainable and cost effective. Finally, next generation pharmacopoeia. I'm very interested. We are working on some of the projects where we are trying to take the compounds like carotenoids like fucoxanthin, astaxanthin, beta carotene, and trying to look upon how it is affecting the anti-inflammatory activities of TB. It is involved in neurodegenerative diseases like Parkinson's, and also it was working in cancer. So there are different pharmaceutical, cosmeceutical, and therapeutic, and this is gaining large and a huge market of therapeutic proteins has been established by Sapphire Industries in USA, where they work on therapeutic proteins for production of therapeutic proteins. And this is very important source where this is, and the pharmaceutical, nutraceutical, and cosmeceutical. So there is a need of our where you can produce these multiple products at a time. And the next generation farmer, you are the people who can decide how you can formulate these different compounds in order to make natural products, and there is no cross reactivity. So it is very important to understand, and even it is very relevant that using these algal byproducts, we can treat number of diseases in a natural way, which does not have any side effects. So with this, lastly, I would like to thank my group, Omics of Algae group. We can see I have an international PhD student. Two of these my students, most of work is presented by these Mukul and Ikras, and they've already submitted and will be awarding soon. And this one of my postdoc is leaving to abroad for his postdoc, and 4 people will be submitting this year. So this is my small group where we are trying to understand. And first of all, last, I would like to thank our funding agency, DBT, DST, BIRAC, Mission Innovation, and to do this fascinating project. We know that DBT has funded us almost 50 to 60 crores for last 10 years, and we were successful in demonstrating how we can show the better biofuel sector, which was a pioneer in one of the areas of research. So we thank the funding agency for always support, and finally our director, Icgb, and all our administrative staff, AO and other guys, who was always instrumental in helping us to get funds and extramural grants. And without our students, finally, I would like to thank my students. Without them, this kind of work cannot be fulfilled. Thank you very much for your patient listening, and I'm open for the questions. Thank you.
Over to Namira for questionnaire.
Good afternoon, sir. It's a very excellent lecture, sir. And we got more questions, nearly 10 to 15 questions we got. So the first question I want to ask you, sir. So what is the molecular system of algal biofuel? Is it similar to petroleum products?
Yes, basically what we are trying to look is that the composition of microalgal is equivalent to biodiesel. So that is what it is equivalent to petroleum products. Only thing is, once we have the basic, like we can transesterified into either FAMEs, or we can distillize into different petroleum products. So basically this is the same thing. That's why we call it as equivalent to fossil fuels, that is petroleum products.
What is the content in algae which is responsible for generation of biofuel?
It is mainly the lipids, neutral lipids. So basically algae, microalgae can produce large amounts of neutral lipids. That is what it is used as biofuel precursors. And macroalgae can produce carbohydrates, which can be used as a bioethanol.
Next, sir. Transgenic plants are not well accepted in India, except BT
Yes.
cotton is available. So what is the status of acceptance of transgenic algae in Indian market?
At present, no, nothing, because only few countries have accepted transgenic CRISPR-based microalgae as safe organisms to do open pond or closed pond reaction. But India, there is nothing. There are developments coming on with the transgenic. That's why I told you in my lecture also, it is important not to manipulate genetically, but also to understand what is happening naturally in the habitat, natural habitat, so that you can fine tune with nutrients or that kind of chemical modulators that is not toxic to the environment, and it can go ahead. So there are both ways. So we have to be prepared with both the answers. The other genetically modified, well and good, it can be done in photobioreactors. If it is an open pond, you should have something where you can just do media engineering, change the composition of the media in order to get those things.
Okay, sir. So with coral bleaching in waters of the world as a major challenge,
Yes.
would it affect the scale up of algae-based biofuel production? And same with the fossil fuels also, because both are the natural resources.
Yes.
So, and same thing, if during your work, if you have faced any issues during this regarding to this coral bleaching?
So basically, coral bleaching is majorly the cyanobacterial toxins which are being generated. As I told in my lecture also, most of the coral bleaching are because of high amounts of toxins which are produced. But if they're controlled at the initial level, it will not be a problem. So basically, the coral bleaching happens. This is way how you can enhance, because in the natural habitat, the coral bleaching is the basically the biomass enhancement of the because there are a lot of nutrients available, but it does not affect anything to do with the fossil fuels, because fossil fuels remains in the depth of the sea where millions of years ago this has been happened. So basically, it does not affect any of these what you call is algal biofuel production, and basically these are not directly grown in the sea. So there is a shore project which is happening where they're trying to grow these algae in a contained sea environment so that you can have the control conditions. But you cannot just throw anywhere in the sea, because you have oceans and trees, and you can do that because you have to collect that, and in order to produce the products, it will not be happening. So it is like you have to have contained facility so that you can grow that and get the benefit out of that.
Okay, sir. Sir, marine water or a freshwater algae, which is more preferred, sir, for the production of bioproducts?
So algae, basically algae marine, it may be irrespective. Marine has more of products are being used. Example, fucoxanthin, astaxanthin, and all those things are from marine this thing. And it does not depend on what kind of algae, and algae has to be characterized what kind of product it is producing. For example, we work on Monorephidium, which produce tocopherols. We work on Chlorella, which produces lutein. We work on astaxanthin producing strain. So there are different strains which are produced. It does not depend, but definitely if it is fresh water, it will be a problem of water source. So we should use wastewater, which is equivalent to fresh water, by cleaning up those wastewater. And fortunately, you can clean up that wastewater to make the water potable.
Similar, sir, if we are cultivating in fresh or marine water, is there any change in their nutrient content or toxicity?
Yeah. So basically, you are trying to change the media of the freshwater to marine. There will be changes, but marine to freshwater, you will not have any huge differences. Marine can easily sustain in freshwater, but freshwater need to be adopted to marine in order to go ahead with that.
Okay, sir. Sir, throw some limelight on commercial viability of algal biofuel, by when they are expected to achieve the commercial viability.
So that all depends on your young, bright minds. So how you can take it up to the next level. But as I said, we are looking for next 50 years. That is not a problem for today. So by 2050, we are expecting that we'll have a great solution for commercial viability of biofuels. Already we have a roadmap. The NREL, as last slide, which represents the roadmap, the feasibility is there, but commercial viability will come in due course of time. So people are working on the cost economics. So they are trying to produce high value products so that they can invest the cost into the biofuels. So it's, as I said, like there are companies which are very much welcome to work on that, and it may not go to 2050 also, but it is a problem of tomorrow. So it has to be, we are sorting it out in a way, and it is definitely feasible, no doubt about that.
Sir, if we are growing the algae naturally, if any other media content is there which we can enhance the yield of algae?
Yes. So basically what we do is, for example, you want to grow in open ponds and you see the water composition of your, so there will be only requirement of nitrogen or phosphate. So it's just like an agriculture. So algal farming is nothing but agriculture. So it needs NPK for the growth. So you need to add that minor ingredients along with what is required, and you can enhance that. So that's what I told, like media engineering approach through carbon supplementation. So environmental, so there are a lot of flue gases. You can, so the challenge is how to capture that flue gas, what we call it as carbon capture and storage mechanism, and that can be pumped into the algal ponds and it can be taken up. So if the industries can develop a mechanism where they can generate the flue gases into the algal pond or divert into the algal pond, algal can take up as much as CO2 it can, and it can produce excellent products of our relevance. No doubt about it. Yes.
Sir, while explaining about this algal biofuel production advantages, you have told that it is producing the related to electricity. You have told about photovoltaic cells, sir. So as
Yes.
we know that photovoltaic cell is used as a detectors in HPLCs and UV and all. Can we use,
Yes.
sir, for that? This one?
Presently, the bioelectricity phase is in a very big thing, but you can use this photoelectrodes as that alternative for that, definitely.
Oh, okay, sir. And any, sir, while this algal production, sir, while any side effects are there, or we have to face any challenges during this, sir, biofuel production?
Yes. So basically, when you are growing in open ponds, there are problems of brachios and other organisms because these are very nutritious, and the predators come and try to feed on those, and cross contamination. There are issues definitely at large scale, because I'm talking about acres of land where it has to be cultivated, and so that has been dominantly worked out by different companies in due course of time. Yeah.
Thank you, sir. Further, if any questions are there, we'll keep a mail.
Okay, sure.
Now I request Principal Sir to speak few words related to this session, sir.
I think no words are enough to describe his perfection in taking the lecture. I think thank you so much, sir. And you have covered right from the scratch to the top outcome. I think we are very happy to have such a beautiful speaker presenting in our forum. So thank you so much for your wonderful time. And that speaks about your perfection, sir. Thank you so much, sir.
Thank you very much. And once again, I would like to thank organization committee, especially Principal Sir and Dr. Vanita, Dr. Tiwari, for giving us an opportunity. So this is how, research can be enlightened to the students, and being a brains or tomorrow future, you guys have to think how you can take it up. We have trying to establish certain pathway for you. Definitely, it will be a great help. And as I told, anytime you are interested to work on any of these things, we are also, I didn't discuss many projects what we are working. We are also trying to work on some products, like for example, you have drugs in pharmacopoeia, paracetamol, how it is broken down by algae, and it makes that paracetamol ingredients into useful compounds by algae. So how these are things can be sequestered by algae and all, also we are working in collaboration. So that is a beauty of that, and it has multifaceted applications. Especially there was a question on bioelectricity, biohydrogen. So you name it, it can be. That's why we call it as a green gold, unexploited green gold of any country. So that's how it is.
So one thing I want to ask, sir.
Yeah.
Actually, during my PhD interview, I have planned the work on metabolomics, sir. But at the time of interview session, panel member told that it is difficult to perform work on metabolomics. Is it really a difficult, sir? So that I have changed my work actually now to bioanalytical.
Okay. So it depends on the problem, what you're working. As I said, like your experimental design is very important, and nothing is difficult. I think you should have a will, and you can just definitely get it. Maybe you have a facility, you don't have a facility, that is a secondary thing. But as I said, like these are very expensive. For example, you want to do sequencing, you want to do transcriptomics. Transcriptomics is now cheaper. Proteomics is very expensive, but you should have. But not see, my lab itself works on different omic approach. You can understand how much different variety of work. And if you take any lab, only one omics they do. Either they do transcriptomic, proteomic, and also I do a multidisciplinary. So it is. So if you want any kind of assistance or any help in that way, we can definitely, your boss can communicate to us, and I am happy to help you in this case. Definitely.
Thank you so much, sir. Thank you, sir.
Yeah.
So myself, Namira Jubin, Assistant Professor in the Department of Pharmaceutical Analysis, VIPER. It gave me an immense pleasure to propose vote of thanks for the day 2 session of FDP on recent advances in science and technology. On behalf of VIPER, I would like to thank Dr. Pawan sir for delivering a starting lecture on cell factories, new frontiers and innovations in era of industrial biotechnology, which gives us a good mastery in algal biotechnology. Thank you, sir. We will be very grateful to you and hope to see you in such programs in future.
Oh, thank you very much. I would love to visit to Hyderabad. It has been almost many days. I'm from Andhra actually, so I'm from I'm from that place. So I would like to visit your place definitely. And thank you very much again.
Sure,
Thank
sir.
you. Beautiful.
Yes, sir.
Thank you
Yeah.
very much, sir.
I thank to management for trusting us and always brace us to conduct such events. I thank to our principal, Dr. A. Ramesh, who always behind us, motivate us to diminutive such programs. Thanks to Dr. Vanita and team for taking an initiative to take this FDP happen. Thanks to Dr. Santosha for giving a nice introduction of Dr. Pawan. Thanks to all the network and supporting team who always plays a major role for success of any event. Thanks to all faculty and students of VIPER for participating in the events, and a peculiar thanks to all the delegates who have joined the session. And a feedback link will be shared. Okay, you will get a certificate by filling that feedback. I now hope we will meet tomorrow with a lecture by Dr. Gopinath Haldar with more zeal and keenness. I request everyone to join the session 10 minutes prior. Thank you all. Have a great day.
Thank you.
Thank you. Thank you, Namita. Thank you, sir. Thank you, Dr.
Thank you.
Pawan.